like the theme is to follow up on the fantastic use, really, of microbeam technology to study the biology of radiation effects. And particularly, Charles has covered on the bystander effect, and this morning, I believe, obviously, David has covered on the biology that we learn from microbeam technology in order to understand the health risks of ionizing radiation. Not only really the health risks, the health risks are going to come after we understand the biological response of cells exposed to radiation. And one of the main aspects that concern us very much is radiation carcinogenesis. And of course, cancer, as we know, is stochastic. It's a late effect, appears 5 years to 40 years after exposure to radiation. And for cancer, we know that there is really no dose threshold or a very low dose threshold. But the severity of the response when it comes to cancer, it is not dose related. It's not dose related. The probability of occurrence increases with dose, but when it comes to radiation protection, a topic on which Dr. Brenner has spoken and lectured a lot, regulatory agencies, advisory agencies tell us about the linear no threshold model. It is the accepted practice in radiation protection. And for years also we have been taught that the nucleus is a prerequisite to produce genetic damage or an important biological response. And Charles just mentioned to us what is the lesion to trigger a bystander effect. We need to understand that we hit the nucleus, we hit the cytoplasm, but we need to hit sometimes the medium to see, or not to hit a cell, see an effect that may come from the extracellular matrix. But that the nucleus is the prerequisite to produce genetic damage, of course, that came from the initial experiment by Monroe with the polonium needle. As you know, he put polonium on the needle, he would move the needle. If he would hit the nucleus of the cell with a few alpha particle hits, the cell would die. The endpoint used to be cell survival. However, if he would pull the needle away and deposit a massive dose of radiation in the cytoplasm, the cells did not die. So from that time and before that, the nucleus is the prerequisite to produce genetic damage. And the genetic damage could be through a direct hit to the DNA or through the indirect mode of action of ionizing radiation through the radiolysis of water. However, the work, and I am of course covering up for Dr. Hay, who couldn't be here today, and so we will see the relevance of that, the non-nuclear hit, but also that we needed to hit the cells and the basis how, to hit the nucleus of the cell, that we had targeted effect for us to calculate, then to try to assume what the risk would be, the health risk of exposure to radiation. And so we know from Hiroshima and Nagasaki that the people that were exposed to a rather high dose of radiation ended up having cancer. However, for doses below 100 millisievert, it always remained a question mark. Until today, it is still a question mark. If non-targeted effects of radiation would exist, so it has been assumed that this question mark, that the cells that don't receive a nuclear hit, receive a cytoplasm, or as Charles has introduced to us, that even cells that did not see radiation, we have seen P21-WAF1 induction in them, that the risk might be supralinear. Of course, some people believe that we have a threshold, and today, after Steve took us to the different facilities, and that we can irradiate with protons, we can irradiate with low LET radiation, you know, soft X-ray, 4.5 keV, where the LET is on the lower side, and maybe we can irradiate with focused X-rays at lower LET than the 4.5 keV X-rays. So, of course, and I didn't have the curve here. What is the real shape of the response? Some of us may believe that we can, a little small dose of low LET radiation may actually upregulate a protective mechanism that will protect the hit cell, and maybe, as George Iliakis and Munira Kadim have published, that we can have propagation of a protective effect. But that's where the value of the microbeam comes for us to try to resolve these very important issues in radiation protection. And the microbeam is a technology like no other one that will help us to resolve these very important aspects. And the work really to which Dr. Hay has done tremendous input, that cytoplasmic irradiation now, and using the microbeam, that we can deposit alpha particles, helium, ions outside the cell, and he went on to study the biological response when he hits only the cytoplasm with the Columbia microbeam. And this I don't need to introduce. It has been covered, of course, extensively, that the Columbia microbeam is a fast microbeam and getting faster. And the beam spot, as Gerhard had told us, can be as little as 1 micron, and my understanding, the development that are occurring at Columbia with the team right in here, that we are going to a much, much smaller beam spot that will go up to the submicron level, up to 70, 80 nanometer level. And like Charles also mentioned, it's very important that we can have a tissue culture system developed whereby we recover the cells that have been exposed to the radiation, whether through the cytoplasm or the nucleus, and by using the tissue culture strategies that have been developed right here, we can recover over 98% of the cells from the dish to study, in this case, whole population studies. And the really is a landmark in 1999 when an experiment was done here by Li Junwu. At the time he was a student of Dr. Hay. He deposited, the radiation was deposited specifically in the cytoplasm. They avoided the nucleus altogether, and to do that they had stained the cells with hops that will go into the nucleus, and then with Nile red that will remain specifically in the cytoplasm, so they can distinguish the nucleus and cytoplasm. And they went on to look at DNA mutation, genomic mutation in genomic DNA, and they looked at the cells that was introduced by David, the Isabel cells, and they looked at the CD59 mutations on the CD59 locus. These cells have 1 human chromosome, chromosome 11 in them, and the mutation assay is very well developed. So when they deposited 1 alpha, that was the mutation frequency, and then they went on to deposit either 4 alphas, 6 alphas, or 16, 18 alphas. What was quite surprising, that the effect plateaued after exposure to 2 alphas, which is quite remarkable and surprising, because the effect, if we deposit into the nucleus, what I understand from Dr. Hay, that the effect for mutation is dose dependent. Here with cytoplasmic irradiation, it became dose independent. And again, this is a landmark study because with those cytoplasmic irradiations, the cells did not die, but yet they underwent genomic DNA mutations. So studies really follow up on these studies is very critical. We can make a huge input using the microbeam technology. And this, it has been explained to us that the A sub L cells have a single human chromosome 11, and it's at this locus, so they can study mutations in here. I presume this would be the human chromosome 11, and the locus, it's on the on this arm of the chromosome. And when the Lejeune went on to look at the nature of the mutations, he found that after cytoplasmic irradiation, he observed small deletions, versus if it was nuclear targeting, it was large deletions. So the nature of the mutation after cytoplasmic irradiation was like spontaneous mutations, which is very informative from the biology point of view that small deletions arise from oxidative stress. And that in here, really they confirmed that oxidative stress was involved. They went to look using DCFDA. These are dyes that will recognize reactive oxygen species, and they found that in the cells that had cytoplasmic irradiation, there were elevated level of reactive oxygen species. You see here the slight yellowish staining versus the control cells that did not receive any radiation at all, which is consistent that the small deletions come from a reactive oxygen species. So that puts us to think about the mechanism that has been involved. And I think that there is a lot of opportunities for us to contribute to this area in here. How did the reactive oxygen species, so they made their way to nuclear DNA to induce mutation, or that cytoplasmic irradiation, we hit mitochondria, maybe we activated oxidases. And in the cytoplasmic irradiation, obviously, we went through the plasma membrane as well, and we should not forget that at all, because that's where we can be activating some of the plasma membrane-bound oxidases, and ADPH oxidase being one of them. And here, again, the experiment from Lejeune that to confirm the involvement of redox-modulated events looked at the CD59 mutation. The cells received 4 alpha particle. If he treats with the hydroxyl radical scavenger, DMSO, the mutation frequency was totally reduced. If he treats the cells with butyramine sulfoxamine, a non-toxic dose of 10 micromolar, butyramine sulfoxamine is going to deplete the cells of glutathione. That's a powerful antioxidant. You see the mutation frequency, it increased tremendously. And this is here, the background mutation frequency from only 8% DMSO or 10 micromolar BSO. So the idea in here that, and the work in Dr. Hayes' lab follows now continuing on the mechanism of genotoxicity induced by cytoplasmic irradiation, which is really that when we hit the cytoplasm, we can hit maybe mitochondria, we're going through the plasma membrane, we activate an ADPH oxidase, we can activate nitric oxide synthesis, an ADPH oxidase will produce the superoxide radical, NO comes, we get them, they react together to produce peroxynitrite, which will lead to lipid peroxidation. Maybe lipid peroxidation is going and work done. Geraldine and Manuela have discussed these issues in their papers, may lead to the inactivation of DNA repair proteins, so we downregulate the endogenous DNA repair activity of the cell, or that is correct, or the nitric oxide is longer lived, has a longer range, can go into the nucleus to induce DNA damage. And that Dr. Hay, a lot of work done by many of the students, they looked at 8-hydroxydeoxyguanosine base damage, oxidative base damage. But it's an area that is really one will see a lot of advancement in this area, contribution from all of us in here by doing microbeam experiments. And here they looked, of course, they went on and they looked at 8-hydroxydeoxyguanosine, and they find in the cells, they see an increase in this type of base damage. So the base damage in here is also, of course, is indicative of oxidative stress coming probably from perturbation in oxidative metabolism. And that's the level after when you hit the cytoplasm with 8 alpha particles. These will not kill the cell. The cells will remain alive. And if you treat with 8% DMSO, the level of staining for 8-hydroxydeoxyguanosine comes back to almost control level. So, and again, they went on to look for 4-HNE, 4-hydroxynonenal, a marker of lipid peroxidation, and they saw beautiful effects. You see the staining for lipid peroxidation. If they treat the cell with beta-hydroxytoluene, which prevents lipid peroxidation, the effect is very much attenuated. So it is pointing altogether that cytoplasmic irradiation is inducing this oxidative stress that will lead to genomic DNA damage. And I'm sure maybe as you sit, many ideas come into your mind of what type of experiments you might do to follow up in here, and hopefully we can have a chance to talk about that. And it's not only reactive oxygen species. Here, of course, it's another indication that peroxynitrite and ions are involved in this effect on cytoplasmic irradiation, triggering nuclear DNA mutation, hitting the cytoplasm with 8 alphas. The mutant frequency is quite high. If they treat with a scavenger of nitric oxide, the effect is very much reduced. And this one is just to, it's another form of this scavenger of nitric oxide, but it is ineffective. So you can see when they treated the cells with this ineffective scavenger of nitric oxide, the mutation frequency was still high. However, with the effective scavenger, it really came down, really pointing down that nitric oxide is also involved in the effect. And we have a lot of evidence for radiation-induced non-targeted effects, like Dr. Brenner had shown us, and Charles did show us, either through mutation, clonogenic survival, neoplastic transformation, gamma H2X, foci, micronuclei. Micronuclei are very attractive because it's a marker of DNA double-strand break, but we can score many cells quite fast. So it's a very attractive marker intracellular oxidant level. We have evidence for radiation-induced non-targeted effects. We can use monolayer cultures, hit them for mutation studies. One would say, how would we do these studies? Of course, the cells that will receive nuclear hit through nuclear hits, we can target them with 20, 30 alphas, and like Charles told us, a single alpha hit could be from 10 to 20, 30 centigrade, depending on the thickness of the cell, that these cells would die that receive the 20, 30 alphas, and the rest are bystanders, and we can do mutation studies to measure the mutation frequency, and thus work, a lot of it has been done by Hong Ning in Dr. Hayes' lab, and there are different protocols, of course, the medium transfer, and the strategy that Charles mentioned worked out with Gerhardt using the double mylar dish, and they found that there is no medium transfer effect. You really had to hit cells within the monolayer in here and look at effects on the bystanders that are nearby, maybe, but obviously there are many other people that have seen a medium transfer bystander effects, that it could be a dilution, could be effect, could be also the phenotype of the cell, like Charles said. Not all the cells, the experiments done with Brian, they don't all respond similarly. Some cells may respond, some cells may not respond, depending where they are in the cell cycle. What is their phenotype? Their antioxidant potential? All of these come and play a role. So if cytoplasmic irradiation induces nuclear DNA damage, if we do cytoplasmic irradiation, can we induce a bystander mutation, mutagenesis? And that would be, again, another support to non-targeted effects. It's something that is with very important consequences. And this is the work, in this case, done by the Gray lab, Microbeam, in Kevin Price's lab by Chun and Shao, where they irradiated cells through the cytoplasm with 1 alpha or 10 alpha, and they went on to measure micronucleus formation in bystander cells, and they saw, after cytoplasmic irradiation, quite a significant bystander effect. The bystanders also showed micronuclei, that's a form of DNA double-strand break. But if they treated the cells with this is the scavenger of nitric oxide. The NMMA was an inhibitor of nitric oxide synthase, I apologize. With the carboxy PTIO is a scavenger of nitric oxide. They attenuated the bystander effect using these glioblastoma cells. So cytoplasmic irradiation, there is evidence that it induces a bystander effect. And work done here at the Microbeam by Dr. Hayes' group showed, again, similar thing by measuring mutation frequency in the bystanders, and they see a significant bystander effect, whether they irradiate. This is here irradiating all the cells through the cytoplasm with 8 alphas. They saw a significant, they see CD59 mutation. If they irradiate only 20% of the cells, they see the effect. It's almost when all the cells see a hit through the cytoplasm. And that's what they would expect if there were no bystander effect, the level of mutation. And obviously work that continues in Dr. Hayes' lab with Hong Ning, Vladimir, and the whole team is looking at the mechanism that would be involved in both the bystander effect in this slide, whether it's due to nuclear hit or cytoplasmic hit. And as we know, we can have, there is a role of cytokines, role on mitochondria is involved, and plasma membrane mediated events are involved into bystander signaling. The work done at Columbia University focuses a lot on the role of cyclooxygenase 2, and of course work that we do focuses on the role of gap junction communication, direct intercellular communication. And so it's an exciting area that is teaching us a lot, not only about, it's teaching us about intercellular communication under stress, which is also enhancing our understanding of intercellular communication under normal conditions. The idea that, okay, we see hit through the cytoplasm, induces genomic DNA damage, it induces a bystander effect. How about the progeny of bystander cells that when the hit cells receive only a hit through the cytoplasm, and work done by another postdoc in the group, by Burong Hu, had shown that targeted cytoplasmic irradiation induced chromosomally unstable clones. Chromosomally unstable clones, that the progeny of the cells, the bystanders, that existed in vicinity of cells that received the hit through the cytoplasm, demonstrated genomic rearrangements. And this is an example of 3 clones. They define chromosomal instability as it is a clone derived from a single cell that shows at least 3 distinct metaphase subpopulations involving rearrangements of the human chromosome in the asabel cells, and that these rearrangements account for a minimum of 5% of the total metaphase scored. And you can see the 3 clones had shown a highly significant chromosomal instability. So really, in conclusion, that the development of microbeam technology has empowered a paradigm shift in our understanding of basic radiobiology, and from cytoplasmic damage to non-targeted effects and to genomic instability among progeny of non-hit cells, the availability of microbeam technology has offered unparalleled research opportunities. We hope today that Gerhard can tell us what we can hit the mitochondria with high precision and see the signaling coming from the mitochondria. And so it's really, we moved beyond the transition from 2D to 3D human tissues. Plants and the whole organism can be exposed to microbeam, and we gained a lot of mechanistic insight on non-targeted effects, which has a lot of implication not only for radiation protection but also for radiotherapy. After we expose, as we diagnose cancer at an earlier age in children, we can gain an understanding on the emergence of second cancers that might be related beyond the scattered radiation, could be mediated via those non-targeted effects, bystander effects. But really the most important, away from everything, non-targeted effects are basic to understanding the stress response, to understand intercellular communication.